relating to MS, but will be of interest to the audience. So I would like to remind the group that we will record the session to post on our International Women in MS website, so people who are interested in looking afterwards or recommending to watch a specific session can do that. People who are not presenting will be muted during the presentation, so we don't have background noise. And you can use the chat to ask questions as the meeting goes. And we will let people talk depending, ask questions in case the chat is not sufficient. If in the future you're talking about what you've heard today, please make sure you acknowledge the presenters and the entities if you're citing the data. So the goal of the meeting today, as our meetings since March, have been to have open and timely discussions regarding COVID-19 pandemic and multiple sclerosis or related disorders, and share information as it becomes available. So we've had a break in the past months because many people were on vacation during the months of August. We're resuming our calls, and we have two great speakers today. So Dr. Grifoni will talk to you about something not MS related, but of very high interest. And Dr. Zakaria will talk about COVID-19 and MS in Egypt. So Dr. Grifoni, you can grab the screen if you want to share your slides. And we can unmute you. We'll unmute you as well.
Hello, can you hear me?
Yes.
Okay, perfect. So I would really like to thank you for giving me the opportunity to present our work here in this session. And I would like to stress out the word "our" because this is a team effort actually. And the reason why we managed to progress in our science so fast is because there is highly collaboration across different labs. And with that, I would like to thank you, particularly Daniela Weskov, Shane Crowley, and Alessandro Seppe that are coordinating this effort with me. And to show you our science, I would like to make a step back and bring you at the beginning of the epidemics, where a very pressing question was floating around whether we were able to detect an immune response against SARS-CoV-2, as that was a fundamental and key question in order to design an effective vaccination. But, okay. Sorry about that, there is some delay. So what do we intend when we talk about develop immune response in the case of COVID-19? So there are different components of the immune system that play a role in controlling the infection. You might be quite familiar with the B cells component because those are the ones that produce antibodies that usually bind on the surface of the virus and neutralize it. So it prevents the virus to enter inside the cells. So this is a very important component of the immune response. However, in most cases, it's not sufficient to actually prevent the virus to infect the cells. So that's where T cells actually start playing a role. And in particular, the CD8 T cells or killer cells are the ones that are able to recognize if the cell is infected, and they're able to eliminate that. And another component of the T cell immune response is the CD4 T cells or helper T cells. So those cells are the ones that actually coordinate the immune system. So they talk with the B cells in order to produce antibodies, and also they decide the quality of the response that is available during an infection. And actually that really changes whether you're looking at a viral infection or bacterial infection or even during an allergic reaction. So the first question we were wondering is whether we were able to see these responses in COVID-19 patients. And we decided to look into donors that have a mild disease in convalescent phase, because that was representing the immune response of the general population. We also performed some studies in the hospitalized donors, but I'm not going to go into that today. So in the context of the COVID-19 convalescent donors, the good news is that we were able to see a very strong response in the 3 compartments that are determinant for an effective immune response. And that the response was significantly higher in our COVID-19 patient when compared with unexposed donors. So that was a generalized response against the entire virus, but as many of you know, the virus is composed by different proteins, and particularly the T cells are able to recognize different proteins of this virus. So we were interested in understanding whether the target of the protein recognized by these T cells was targeted against the spike protein. That is because spike is the main target of vaccination. And indeed, we were excited to see that we observe a very strong response against the spike protein for both helper and killer T cells. But we were also able to detect reactivity against other different proteins of this virus, therefore suggesting that these additional candidates can be added in future vaccine trial. Another point that emerged from our study was the fact that we did see some reactivity in donors unexposed. So to perform a very clear control with the COVID-19 patient, we decided to go back in our blood collection samples, and we decided to study donors that have been collecting in the 2015-2018 range, because we want to make sure that indeed those donors have never seen the virus. So it was pretty surprising for us to see that roughly 50% of these donors were able to actually recognize SARS-CoV-2 peptides as well. So we hypothesized that that was due to the fact that actually there was a pre-existing immunity due to the exposure of other human common cold coronaviruses. And this point was actually interesting, as we know that these human common cold coronavirus are circulating every year, and actually different strain of HCOV are actually present depending on the years. So one of the questions that we wanted to address is, we hypothesized that the human common cold corona can influence the reactivity against SARS-CoV-2. Is that true? Indeed, and how much this pre-exposure can influence reactivity. So to address this question, and that is a follow-up study that we recently published in Science, and Jose Mateo's here picture is the first author of the study. So up to now in our cell study, we were looking at the entire response for the proteins, but to really fine-tuning and understanding the relation with the common cold coronaviruses, we have to reach the peptide levels, so the smaller portion of the protein that is specifically recognized by our T cells. So we did that for our unexposed donors, and then once we have the sequence of the SARS-CoV-2, we also determine the similarity with the human common cold coronaviruses. And as you can see in this graph, when these peptides are recognized by multiple individuals, those sequences have actually a higher similarity with common cold coronavirus, and that is significantly higher than when you look into peptides that do not retrieve response or just response in one donor only. So that indeed suggested that there was implication that the human common cold coronavirus could influence this response. But we wanted to really dig into this and make sure that that was the case. So what we did was to expand these T cells with the specific SARS-CoV-2 peptides, and then test the equivalent version of the sequence for the human common cold coronaviruses. And our expectation was that if indeed the two were correlated, we would be able to see response for both. And that was actually the case. In fact, we do detect a response for SARS-CoV-2, but also the corresponding sequence derived from human common coronaviruses. This specific case, actually, the sequence is very similar. You only have one amino acid of difference. But we look not only on this example, but multiple examples. And in some cases, we were happy to see that actually the response against the human common cold coronaviruses is stronger than the one observed in SARS-CoV-2. Therefore, suggesting that that is the primary virus they've been exposed to, and then the response to SARS-CoV-2 is a cross-recognition. To further validate this point, and this is a very complicated slide, but I just want to give you the main point out of it. The response to be a response that has been there previously, that should belong to the memory response of our T cells. To determine the memory response of our T cells, we use the expression of two different markers. And just as you can see, the marker over here, so CCR7 negative and CD45 array positive and negative, represent the population of effector memory. And indeed, when we compare the response against either non-spike proteins or spike proteins, you could appreciate that the majority of the response, both in red and blue, belong to the effector memory compartment. Therefore, suggesting that indeed this cross-recognition we observe comes from a memory T cells and is not a newly induced response due to the technical approach that we utilized. So in conclusion, we did determine a response in our COVID-19 patients for the three compartments, but we also be able to observe a response in unexposed individuals. And that has been something that we first saw in our cohort, but it was also observed in cohorts all over the world, coming from Germany, the group of Thiel, as well as in Singapore by Bertoletti and other collaborators in Netherlands. And more papers are pointing out toward the same direction. Our hypothesis that this cross-recognition was due to exposure to common cold coronaviruses is now validated with these follow-up studies. And the question that follow up now, and we're still investigating, is does this immune reactivity is able to influence immunity against SARS-CoV-2? So clearly, the fact that you have a memory response suggests that you're gonna respond faster to this virus. Whether this can be detrimental or not, we don't know yet. However, we know in the past there have been cases for other viruses where previous exposure with one virus protect people. That's the case, for instance, of the swine flu, where people pre-exposed, actually older age, were able to be protected by the infection. In the case of common cold coronaviruses, we know that there is a higher exposure in younger age, so that might justify why the age range of protection that we observe can be younger than the swine flu. So what are the implications for the vaccine development in this point? Of course, it's important now to determine whether there is a pre-existing immune response against the human common cold coronaviruses, because if you don't detect that one, you will not know if the vaccination triggered another response, so you're just detecting something that was already there in the baseline. So with that one, I would like to thank you for your attention. I'm happy to take any question, and thank you again for inviting me.
Well, thank you very much for presenting your data. So for people who may not have seen that on the slide, these were data also from your Cell paper published recently. So people can ask questions in the chat or unmute themselves to ask questions, but I'll start with the first question. Do you think that the immune reactivity could actually explain why some people are asymptomatic but are found to be carriers of the virus?
Yeah, that's an excellent question. So of course, we don't know yet if that's the case, but clearly this points out to the fact that if you have a memory response, you might be able to fast recognize the virus. So this can be one of the reasons that explain the fact that we have that many asymptomatic cases. However, nobody has shown this directly yet. So more studies are needed to address this point.
Okay, so Fabienne, do you want to ask your question?
Hi, thank you, Dr. Grifoni, great talk. I had a question about the antibodies, because you showed us the first schematic, first graph with your, I think it was the RBD spike antibody. And I was wondering whether you saw the same things happening with the antibodies, because obviously the T cells will have an influence on the antibody productions and regulation as well. And I might have missed it, but...
That's an excellent question. Yes, actually we test that in our Unexposed over here, and they are all negative. So you do see significant difference. And that is true for IgG, IgM, and IgA. I just haven't shown the other two classes of IgG, but we study them in our paper.
Okay, thanks.
Of course.
Any other questions here? I know there's some immunologists on the call, so don't be shy, ask your questions. David Baker, do you have questions? Tony Reader, do you have questions? Usually you're pretty vocal.
Yeah. It just, hello, Alba, I never met you, but we just used your paper as an exam paper for a preliminary exam. Okay, it's very important. Okay, and I think that the T cell responses that you're dealing with are something that's been neglected in a lot of the literature. People are talking about COVID antibodies, and how do you weight the relative antibody versus T cell response? And second question, the role of interferons in this picture.
So I think we can't simplify and just focus on one branch of the immune response. Many data have shown that without one or the other one, you might not have an efficient response. It is true that T cells, particularly in the COVID case, are important. There is a nice report with a gamma globulinemia, a patient that got the COVID-19 and were able to control it. So that suggests that actually T cells have importance in this infection, maybe more than other type of viruses. But definitely an antibody response is very important. As I was mentioning at the beginning, you want to be able to mount efficient neutralizing antibody that are able to actually bind to the virus and prevent the infection. And in that context, it is important to take into account the crosstalk between B cells and CD4 T helper cells. So the bottom line is, why focus on one when you can look at the three of them? About your second question, I'm sorry, do you mind reminding me?
The role of interferons in T and B responses and coronavirus. Yeah.
Well, so clearly, and I haven't shown this, but the T cell response that we see are heavily Th1 polarized, therefore it is important. It's also true that the interferons are important in guiding the immune response. In our studies, we focus way more on the adaptive immune response because that's the one you want to trigger in the context of vaccination, but definitely it's a way more complex picture. So it's important also to look into those as well.
So we have a question from Wendy Gilmore. Wendy, do you want to unmute yourself and ask your question?
Sure, great work, Alba. I just wondered if you are doing cytokine secretion analyses on the cells that you're looking at?
Yes, so we did that for our cell paper. We did the CBA assay actually to determine the type of cytokine. And as I was mentioning before, we do identify mostly a response with interferon gamma and IL-2, therefore suggesting that the overall polarization of our T cell response is more toward Th1.
Thank you.
Sure.
David, do you want to ask your question?
Hi, I guess the question I would like to ask is, in terms of the vaccines, what should we really be targeting? Should we just be going against the spike because that's what many of the vaccines do? Or should we be going much broader? What's your opinion on that?
Well, definitely, as we've shown for our T cell response, the stronger target of reactivity is indeed the spike. So that might be sufficient to achieve a protective immune response because in there you have a good B cell response and T cell response. It is also true that in our studies we show additional targets. So I wouldn't exclude the possibility to add those in future vaccine candidates, but I wouldn't say necessarily that just having spike is not enough to achieve protection.
Could I ask one more question? And my question would be, what would be your advice to somebody taking a B cell depleting drug or a B anti cell depleting drug in terms of what we should do in terms of potentially getting a vaccine response in them?
B-cell depleting. Well, as I was mentioning, there is this nice study with the gamma globulin suggesting that the T cells can be sufficient in controlling the infection, although these are people that still develop symptomatology, therefore suggesting that B cells are important. So definitely the spike protein is enough immune target of responses to actually trigger your T cell response, and perhaps I would add additional candidates in this case, in order to have a broader T-cell response against the virus.
So Jeff, do you want to ask your question?
Oh, sure. So beautiful talk. So my question was whether treatment of someone with infection with convalescent plasma, although it may help clear the virus, prevents generation of an immune response against the virus.
So I believe there are some studies that show that, that is an approach that has been used in the context of many infections, and definitely neutralizing antibodies should be able to reduce the the capability to infect. However, I guess for a treatment purpose that might be interesting to pursue, for a vaccine purpose, of course, you want something that is able to trigger a memory response, therefore you want both B and T cells.
Yeah, so specifically my question was not whether it helps clear the infection, but whether it then prevents the generation of an immune response against the virus. So it's a short, it may prevent a long-lived immunity against the virus.
Well, that assuming that the B-cell treatment caused sterilizing immunity, which I'm not sure that is the case in this infection, and I would like to see some data about that. But if you get actually infected, even if the amount of virus that infect the cells is less, then that will be still enough for the antigen presenting cells and all the cells to present the antigen to the T cells to trigger an immune response.
Okay, thanks.
Sure.
Okay, Magd, do you want to ask your question?
Yes, thank you very much, Alba, for this very interesting talk. Actually, will make my presentation afterwards look like bedtime stories, you know. So this is, the question is, we have some indication that the BCG vaccination, which is a T cell response mainly, has some kind of protective role in COVID-19. There has been these papers that those who have been vaccinated with BCG maybe have less severity of the disease and maybe less incidence. Could this have something to do with your? Could this explain? Could your work explain this theory or?
Well, in this study, we have been addressing the question with the human common cold coronaviruses. We are looking into that as well. And up to now, I can't say neither yes or no, because we're trying to look into that. It is true that behind the human common cold coronaviruses, there might be other players that can actually contribute to the pre-existing T-cell immune response, and we're still looking into that. But yeah, definitely that would be an interesting point to address in the future.
Thank you.
Sure.
Any other questions? Okay, well, thanks very much, Alba, for the presentation. We'll go to Magd now, who's going to talk about COVID-19 and MS in Egypt. So you can grab the screen.
Can you see my screen now?
Yep.
Okay. First of all, thank you very much, Emmanuel, Fabian, and all the organizing members of this beautiful meeting for giving me the opportunity to be among you and share some of our COVID-19 experience in Egypt. You know that Egypt is a low to middle income country with a population of just above 102 million people. And Egypt has seen a lot of development in the field of MS in the last few years. This was a slide I presented with your meeting in April. And this was the beginning of the pandemic in Egypt, where we only had 2,350 cases, and this is in the middle of April. We participated by 2 centers in the multiple sclerosis COVID-19 study, which is led by our dear colleague, Maria P. Sormani. And we participated with 2 Egyptian centers, the MS Ain Shams University Center, which is the largest registry of MS in Egypt with more than 3,552 cases. And the Cairo University Center, which is the second largest with 2,732 cases in the registry. Now, I know that a lot of you maybe don't know that MS exists in Egypt, but just to assure to everyone about the quality of the registry, there have been several publications in Multiple Sclerosis and Related Disorder Journal about the Ain Shams University registry, which I will be talking about the preliminary results of this specific registry. And also in the MS base, among 35 countries, Egypt is ranking number 7 in the patient enrollment by country, and more than 70% of these cases in the MS base are from the Ain Shams University registry, which I will be talking about. Also, we had the cooperation of the MS Care Society, which is the largest, maybe the largest in the Middle East patient society, with more than files of 10,000 patients, and a very professional organization. The founder of this organization, who is Dr. Mai Sha'rawi, is currently the president of the MSIF. Now, COVID-19 has had an impact on patients in Egypt as much as they had all over the world, and all the scheduled visits have canceled, and we had financial implications, and the health services were stuffed and loaded, and patients were confused, and if there were any hospitals working, then they would move from these hospitals to the others. And it was at the beginning, especially at the beginning, it was like a chaos. Everybody was seeking how to communicate with the patients virtually or through telemedicine or communication like phones, WhatsApp, Facebook pages. So these have been the tools of communication with the patients, whether in general to assess their follow-up or whether to assess if they have COVID or not. And so this has been the scheme, is that the patient communicates through either of these media, and then we do the questionnaire, and if there are functional skills, and review the labs and see what are we going to do about these patients. But the important thing I wanted to tell you about Egypt as part of the Middle East is that this kind of telecommunication is very popular and has been there for a long time as part of social activity and as part of direct communication between the physicians and the patients. So we have Facebook pages for nearly all of the patients of MS. We have WhatsApp groups for all of the patients of MS. And at the same time, we have in each center or university or MS unit, we have movie stars, which are physicians, professors or associate professors, who are always involved with the patients in these WhatsApp groups or Facebook pages, nearly 24/7. I don't know really how they have the time to do all of this, but this has made the communication with certain characters in each center very easy and continuous. So at the beginning, patients were reluctant because we had these strict rules, quarantine, for everyone who proved to be a COVID positive patient, and the patients were reluctant and afraid to talk about their symptoms. And then gradually, when things started to open up, then we had the opportunity to communicate with the patients when they had symptoms and then send them the questionnaires, which we wanted to include all of them, all of the data we needed. And this is part of a questionnaire, which is really a collection of several questionnaires in one questionnaire. PCR, symptoms, comorbidities, lymphocytic counts, severity, admission, recovery, additional data, lost EDSS, is there specific symptoms like loss of taste and so on. The peak of the COVID-19 was in Egypt was in June. So we can see here, this is 15 of June, Arabic language, 19 of June. So we have nearly 6,000 patients, more positive COVID-19 within only 40 days. And astonishingly, when the peak came, the government started to open up. There were very strict regulations at the very beginning, but when we had the peak, the curfew was later till 12 o'clock at night, and the hospitals were stuffed with patients. So there is no strict regulations that you have to quarantine the patients. You can go and quarantine at home, and things, because of probably overload of the system, it started to open up. So our registry in Ain Shams University, as I told you, I will be speaking about this preliminary results. We had collected 78 patients. 68 of them were relapsing remitting, 10 were secondary progressive. Majority were females, 82%, and the majority were mild cases representing 72%, and the severe cases were only 2 cases. Now, because of the problems of the PCR, we depended more on the chest CT. Now, in Egypt, we have a lot of private activity, private systems, and private radiology centers. So those suspected of COVID would just go to a radiology center where they can do a CT chest with less than 50 euros, and they have the typical bilateral, subpleural, ground glass opacities that are characteristic of COVID. So you can see here that only 12 of the 78 performed PCR, while around 34 performed chest CT positive, and 7 in combination of both. We had also 40 cases on clinical grounds with the typical clinical picture of fever with anosmia or loss of taste or other symptomatology. And these are the reported symptoms among the 78 cases, with the main symptom being fever. And those with fever associated symptoms were anosmia more than 50%, and even diarrhea 30%. 35% of these 78 cases had direct contact with probably one of the family members in the house, and 65 did not have contact with with a COVID positive patient. And the CBC collected from the patients showed that more than 56 and 59 showed a decrease in the total lymphocytic count and total leukocytic count and the total lymphocytic count, the absolute lymphocytic count. Now, among the 38 confirmed either by PCR or chest CT, the majority of these cases were mild, second moderate, and only 2 cases were severe. So most of them were mild. Now, here is a very important point. We're not talking about files from hospitals. We do not have, in the quarantine hospitals, we do not have files for MS patients that have been quarantined. This is more like a real world experience where you have direct communication with the patients. But at the same time, because of this a very strong social network, which is in the Middle East, so if there are patients who have been hospitalized, we would have heard about them in the social media, and we didn't. Among the 40 patients who did not perform either chest CT or PCR, again, the majority of the cases were mild cases, 85%. And this is the DMD distribution for the both groups, the 38 cases confirmed and the 40 cases only on clinical grounds. You can see that only 2 cases were severe, one with rituximab, one with fingolimod. These were admitted to hospitals. The rituximab case died. It was a secondary progressive case. EDSS 6.5 have been on rituximab for about 4 or 5 years, and there were comorbidities with this case. And the case with fingolimod recovered, although being severe. And the distribution of the DMTs is more or less the same within the groups, so it doesn't point preliminary to any implication of any specific DMD being involved in the severity of the disease. Again, to tell you about the development of this DMD availability in Egypt, in 2011, we didn't have any DMDs available on the expense of the government, whether Ministry of Health or social insurance or the national insurance. And now we have all these medication. You can see they are available completely free for all of the patients. These are the total cases, the 78 together, where we have the mild, the moderate, and the severe. And again, it's a more or less equal or even distribution. Maybe interferons represent the majority because interferons are the majority of patients being used in Egypt. As for the admission and the recovery, all of the 78, only 1 patient died and the 77 recovered. And we had 1 in the ICU and ventilation, which was the rituximab case, and the rest of the patients were home quarantined. Now, these are preliminary results from just 1 center participating in the MS COVID-19 study. With the help of my dear colleague, Professor Maria Pia Surmani, we will be implementing the results with the other MS center in Egypt, which will collect at least about 120 cases, and they will help us with the detailed specific statistics of these cases. Again, thank God, now we are at least down the hill, or up the hill, I would say. We are in a better condition. These are the new cases, only 103. And up to now, the total number of COVID-infected patients in Egypt is nearly 97,000. So now we are in the downhill of the COVID infection. We hope it doesn't go again up. And finally, again, these are not from the hospital files. Maybe this is a different perspective. This is from direct communication with the patients, but because we have a very strong social network in the Middle East, and to tell you about the social network, there has been a statistics that showed that the number of mobile lines in Egypt is 4 million more than the Egyptian population. That means everybody in Egypt has a mobile phone with WhatsApp and everything, and 4 million have more than 1 mobile phone. So thank you for listening. Thank you.
Thank you very much. So the presentation is open for discussion. So Magd, what do you think is the proportion of patients with MS in Egypt who are on any kind of disease-modifying treatment?
Well, the number of patients we registered on any kind of disease-modifying treatment is nearly 10,000 patients in Egypt. And according to the WHO, we have a problem in epidemiological studies, you know, because we don't know exactly how much patients we have. We see a lot of patients, and the WHO statistics assumes that we are in the range of 50 to 60 per 100,000, so that in a 100 million would make 50,000. And this means that if this is correct, then only 1/5 of them are receiving DMDs. But now we have a very strong social media about MS, and people are knowing more and more about the disease. We have television programs, we have radio programs, and so we expect that by time we can capture the rest of these patients. especially in Upper Egypt. Upper Egypt is the, we call it Upper Egypt, but actually it is the south of Egypt, where you know, near Sudan, where you have more tribal areas. And so they don't receive this much knowledge that there is a disease called MS, and it has medication for free, because now everybody has the right to have free medication in Egypt.
Great. Any questions from the audience? So I don't see any question in the chat. So is there, because sometimes the outreach to patients is limited by the availability of neurologists. Do you have several neurologists in the south of Egypt? Or is there, because it's less densely populated, you tend to have not too many neurologists that can prescribe also these medications?
We have 3 universities in the south of Egypt. Most of the neurologists are university candidates, but we have a lot of neurologists. My university, which is Ain Shams University, my department has 250 neurologists, starting from the professors and ending to the residents. So we have a lot of neurologists, because we have in Egypt around 19 governmental universities, apart from the private universities. So the physicians are there, but they are not distributed all over Egypt. You know, we have certain capital areas in the south of Egypt, like Aswan, if you know Aswan, and like Luxor, okay? Yeah. So you have the physicians there, but all around these areas, you don't have the specialists over there. So if the patient doesn't travel and go to this city, then the service might not be available over there. Of course, this is not the condition in Cairo, you know. In Cairo, we are more than 20 million people. In Cairo.
And then it takes time to travel to the MS clinic because of traffic, but just kidding. So anybody else has questions? Otherwise, I wanted to remind the group that there will be specific sessions of COVID-19 regarding COVID-19 and MS during the late breaking news of ACTRIMS, so MS Virtual 2020. Don't know whether Jeff is still there to tell us more about that. I think he may have dropped. So just wanted to remind the group that there will be very interesting sessions here. I think Etel, are you still on board? Because I'm curious to hear about what's happening in Chile. And I think she may have dropped also. Okay, well, thank you.
David and Tony had a question, Emmanuel.
Oh, sorry,
so it's at the end of the chat.
Tony, yeah, thanks for, Tony, do you want to ask your question?
So I thought that was a very interesting profile in Egypt, and looking at the distribution of the drugs and the people who had COVID, does it reflect the same rate of use in the population in the city, or is it more heavily weighted towards some of the drugs that we see in the COVID sick people versus the unsick people? I don't know if that made sense.
Thank you very much.
About your background use. Yeah.
Thank you for this question, because I will explain that we had interferons only until 2015. And then in 2015, we had fingolimod on the expense of the Ministry of Health and the governmental reimbursement. And only in the last 2 years, we have had DMF, teriflunomide, ocrelizumab, and rituximab. So although these medications are there, but of course the majority of cases in Egypt still are on interferons or fingolimod.
Right, so I'm wondering if there's a disproportionate representation of the non-interferon drugs, or
it's nearly the same, because in these groups we had the majority cases interferon, and second we had fingolimod.
Or the other way around.
And the others were sporadic cases, you know, 1-2 cases, sporadic cases.
Great.
And then there's a question from David Baker. David, you want to ask your question?
Hi, it's just a quick question to ask. Have you been able to test any of the individuals to see if they're seroconverting or not?
Those who have been positive, the 12 cases that have been positive have been negative in the they have been negative in the second or third swab they have done. The only 12 cases.
But that's for virus. I'm asking, do they actually make antibodies?
We hadn't tested. Unfortunately, we hadn't tested. Only one patient who had a high IgM titer. This is one patient only, and this is because of the limitation of the test itself. It's a relatively expensive test in the Egyptian market, and it wasn't available for the public. It was only with the Ministry of Health hospitals, and people were afraid to go there because at the beginning, if they tested positive, then they would take them and quarantine them. So it was they avoided this, and we relied more on the CT chest results more than the PCR results. Now it's open. Now the PCR is open, and you even have to have a certificate of the PCR before you leave Egypt that you have been tested and things like this.
Okay, thank you very much.
Thank you.
Thank you. Okay, so if there is no other question, I will close the session. Thank you very much for your participation. We probably will skip a few weeks of meeting because of ACTRIMS, and then we'll resume our regular calls after MS Virtual 2020. Thank you for your participation.
Thank you very much. Thank you. Bye-bye.